and I'm going to be presenting my poster on analysis of total PFAS concentration in solution. So just to kind of summarize what we're doing, we're looking at per and polyfluoroalkyl substances. These are aliphatic compounds that contain carbon-fluorine bonds. They're really, really strong bonds. So they're highly persistent in the environment since they don't degrade significantly. These compounds have been linked to a lot of health concerns. So it's important to analyze samples in drinking water, for example, to hopefully get an idea of whether the concentration is safe or not. Liquid chromatography mass spectrometry, or LCMS, is a highly sensitive technique that's used, but it's really, really expensive. The other issue with LCMS is that it leads to underrepresentation of total PFAS contents. There's thousands of different PFAS, so you're analyzing thousands of different individual PFAS using LCMS. Our solution to that is to develop a sensor that can measure total PFAS in a sample. This is accomplished by mineralizing a PFAS in a reactor, which produces fluoride ions. This fluoride is measured using a fluoride ion selective electrode, which produces a potentiometric response. The potentiometric response can be plugged into the Nernst equation, which allows us to determine the total concentration of those fluoride ions. Knowing the concentration of those ions allows us to compare it to different PFAS compounds, and then we can get an idea of like the total PFAS contents in a solution. Using our sensor requires a manual 6-port valve, a peristaltic pump, the fluoride ion selective electrode, and then a custom flow cell. So we filled the 6-port, we used the 6-port valve to fill an injection loop. It's 100 microliters, and then we inject that 100 microliter sample into the FICE and flow cell using a carrier solution. We're able to repeat that process in succession, which allows us to analyze multiple samples and look at changes, small changes in potential over time. This is really useful compared to LCMS because we're able to perform this quickly and it's cheaper. Also, just batch-wise analysis, only using the FICE and not using the 6-port valve in the flow cell is time-consuming and tedious. And with the combination of all these parts in that sensor, we're able to observe small changes in real time. It's super useful. So the FICE sensor provides us with efficient means to determine total PFAS concentration in solution post-degradation. The goal is to obtain part per billion or part per trillion level sensitivity when measuring fluoride concentration. Being able to measure super sensitive concentrations like this would be a good substitute for LCMS, which is able to measure it parts per quadrillion, but again, it still has that underrepresentation issue. Our focus remains on PFAS analysis. However, this sensor could be really useful for any other ion analysis that's applicable. So it's got a lot of potential. I think that's it. So thank you guys.